Are we live? Yay, I see myself now. Okay. Hi, welcome everyone. I want to thank you all for coming to another edition of our virtual Tyson Summer Seminar Series. I also really want to thank Tyson itself for providing the resources to make these seminars happen. It's truly a privilege to be able to host scientists from around the country and bring them in to give a seminar. And as a Tyson staff member, and if you're a faculty member that uses Tyson, we get the opportunity to choose a person that we want to bring in and get to know better every year, and that it truly is an honor and a privilege. And so, big shout out to Tyson for making that happen. And even though it's really unfortunate this year that we can't all be as a community physically together, I know that our community remains strong during this time. And if anything else, we're just watching our seminars in more comfortable chairs this year with maybe some better air conditioning than we normally would. So this is not all a loss. So I'm really pleased today to introduce Dr. Megan Meuti. Megan is an insect molecular physiologist and assistant professor at The Ohio State University. Megan has basically done most of her career at The Ohio State University. Must be a town she loves. It's a town that I love, actually. I got the opportunity to spend a weekend trawling around in the alleys of Columbus collecting mosquitoes that were then used in several publications. So that's kind of a cool connection. So as an undergraduate, Megan received degrees in both entomology and microbiology. And she completed her PhD at The Ohio State University under Dr. Denlinger, who is basically like the guru of insect diapause, which is a hibernation trait that insects in temperate climates have. And it's a subject that is really near and dear to both Kim Medley and my heart. We've done a fair amount of work on that subject. So Megan is not only a teacher, an author, and a very productive PI running a great lab. She's also really committed to science outreach, and she can frequently be found bringing live insects into classrooms, which is also something I can get behind. So I first met Megan this fall at the entomology conference, and I noticed her nodding her head a lot in the audience of a session that I ran, and I just thought to myself, like, man, I gotta get to know that woman. She's obviously into the same stuff I am. And it also happened that at the same meeting, one of her graduate students gave a presentation on how artificial light at night impacts diapause in mosquitoes, which is verbatim, basically, what I was talking about at that meeting. So it was just really, serendipitous that we got to meet, and I've been trying to talk her into working with me ever since. So we're very pleased that we have her here today to give a seminar. And even though we can't hear you, please join me in welcoming Megan.
Thank you so much, Katie. You didn't really have to do very much convincing at all to get me to want to work with you. We're kindred spirits, I can tell. And I'm just so honored and delighted to be with you all today to tell you a little bit about my research on mosquito diapause. So that's the seasonal response that I mainly focus on. And just to give you a roadmap of where we're headed today, first I'm going to tell you lots of things about insect diapause, more things than you might ever have wanted to know. And then I'll introduce you to seasonal responses in the mosquito species that I work with, my lab works with, the northern house mosquito, Culex pipiens. Then, as a molecular physiologist, there's going to be a fair amount of molecular physiology here. So I'll talk to you a little bit about the work that we're doing to try to figure out how, at the molecular level, mosquitoes are able to measure day length and tell when winter is coming, and whether or not their circadian clocks are involved in that process. And then after that, I'll tell you a little bit more about some of the more ecologically focused work that my lab is just starting to do in figuring out, how diapause really matters to the seasonal ecology of mosquitoes and disease transmission. And I'll talk to you a little bit about some of our findings on how artificial light at night impacts seasonal responses in mosquitoes. So, yay. Okay, so Mammalian hibernation and insect diapause are similar in a lot of ways. These are both states of arrested development that these animals are entering in order to survive adverse seasonal conditions. And in this case, in temperate environments, it's winter. So both diapausing insects and hibernating mammals will increase their fat stores, and they also both seek out protected locations to hibernate during the winter, aptly named hibernacula. And they will also both upregulate stress responses, things like heat shock proteins, as well as antimicrobial peptides that help them combat pathogen attacks. And in both hibernating mammals and diapausing insects, they reduce their metabolic rate, which is really important because they, spend the fall acquiring fat, and then they're not able to get a lot of food during wintertime. So having a reduced metabolic rate helps them to hold on to those fat stores for as long as possible. And in both cases, these responses are induced by photoperiod or day length. So they're a photoperiodic response. And to convince you of that, I have some data taken from one of my research advisor's papers when he was a PhD student. So here on the y-axis, we have the percent diapause incidence. And on the x-axis, we have the hours of light per day. And this is in the flesh fly Sarcophaga bulata. And you can see that when the days are short, less than 13 hours of light per day, the diapause incidence is quite high. And then once the hours of light per day get above 14, the diapause incidence is basically zero. And it's a pretty precipitous decrease along here. So it really is day length that triggers diapause in insects, the same way that day length also triggers hibernation in mammals. But of course, we also know that mammals and insects are pretty different in a lot of ways, and not surprisingly, this also impacts their overwintering responses. So mammals, of course, are endothermic, and although hibernating mammals have a decrease in their basal body temperatures, it's still generally quite a bit warmer than their surrounding environments. Not so with insects, which are ectothermic. Their body temperatures are always going to be pretty close to their ambient temperatures. And some insects, when they enter diapause, even become freeze tolerant. So you can freeze them solid and then thaw them out later and they're totally fine, which is pretty amazing. Also, because mammals have a very long lifespan and they develop gradually, they hibernate many times throughout their lives. And they can also hibernate in any different stage, as juveniles or adolescents or adults. However, because insects have a very short lifespan, and particularly for those that are multi-bolting or have many generations per year, they might not overwinter at all. And if they do develop, or if they do overwinter, for each species, they're usually only capable of overwintering in one specific life stage. And when we look across insects, we see example of insects that are capable of diapausing in every insect life stage. So for example, the Asian tiger mosquito that Katie and Kim study, Aedes albopictus, overwinter as eggs, technically as ferrate first instar larvae that are hidden away beneath the eggshell, but that's how they spend the winter. The pitcher plant mosquito, Yamaya smithii, actually overwinter as larvae inside of the pitcher plants themselves. That's where they get their name. And the flesh fly Sarcophaga bulata that I just showed you that photoperiodic response curve a few slides back, they overwinter as pupae. And then my mosquito, the northern house mosquito, Culex pipiens, overwinter as adult females. And I'm sure all of you, as very astute biologists, have realized that 3 of these examples are mosquitoes, and the other example is another fly, another dipteran. And this shows us that diapause has evolved multiple times in insects and indeed multiple times among mosquitoes, which is really interesting. Okay, so now I'll tell you a little bit more about seasonal responses in the northern house mosquito, Culex pipiens. This species is the major vector of West Nile virus throughout the northern United States, which is why we can get some funding to study it. And in this species, only females enter diapause in response to short day lengths. And their diapause is characterized by low levels of juvenile hormone, which is the hormone that signals ovarian development in adult mosquitoes. And because there's low levels of this hormone, that means that their eggs don't develop. So they're diverting resources away from reproduction and towards survival. Also, my collaborator Chao Ho Sin has found that there's low levels of insulin signaling and increased levels of the fork-head transcription factor FOXO, which is kind of this master regulator of a bunch of genes associated with diapause. And the end result of this is that they're able to pack on the fat, increase their fat stores, and then also increase their stress tolerance. So the two phenotypic markers that we usually use to study diapause are egg follicle length and fat content. So over here we have a non-diapausing mosquito, and you can see that she's pretty slender. And then if we dissect her ovaries, and this is before she's had a blood meal, we can see that our primary egg follicles are pretty large, usually around 100 micrometers in length. And in contrast, her diapausing sister over here that's been exposed to short day length is pleasantly plump. And when we pull open her ovaries and look at her egg follicles, we see that they're much smaller, usually around 50 to 70 micrometers in length. Okay, so not only is diapause really affecting the kind of reproductive physiology of the mosquitoes, it also affects their behavior and ecology as well. And so just to drive this home, under long day conditions, we see that male and female mosquitoes will emerge from the water and they will get together and they will mate. And then shortly after that, the females do what we hate them for. They bite us, they take our blood, and they use the protein from that blood to produce their eggs, which they lay in beautiful rafts on the surface of the water. However, under short day conditions, male and female mosquitoes again get together and mate. But shortly after that, in the fall, the males die. They do not survive the winter, leaving the stronger sex to prevail. And those females feed, rather than biting people, they are completely disinterested in biting any vertebrates. Instead, they'll feed exclusively on nectar from flowers, and which they can easily convert into their fat stores. And then they'll seek out their hibernacula, including culverts and sheds in people's basement. And that's where they'll spend the winter. And for that whole duration of winter, those 3 to 6 months, those female mosquitoes are keeping the male sperm alive in these 3 sperm storage organs called spermathecae, which is pretty impressive. And then in the increasing temperatures in spring break the females out of diapause so that they'll bite a vertebrate, use the protein from the blood to lay eggs, and unfortunately for us, the cycle of life continues. So one might wonder why I'm so interested in diapause and studying it, and plan to spend most of my career studying insect diapause. And the main reason is because seasonal changes in mosquito abundance are what is really driving seasonal peaks in West Nile virus transmission. So this graph is taken from the Centers of Disease Control, and we can see the number of human cases of West Nile virus on the y-axis and the time of year on the x-axis. And we can see that there is a peak in the number of cases of West Nile in the late summer, early fall, shortly after the time that mosquitoes have transitioned from biting birds to biting people. So if we can really better understand how and why mosquitoes are entering diapause, we could reduce disease transmission, not only kill off the mosquitoes, which would be great, but also reduce disease transmission. And I think it might be possible for us to trick mosquitoes into thinking that it's wintertime when really it's summertime, so that way they don't bite us, which would be amazing. Or perhaps better yet, trick them into thinking that it's summertime when winter's on its way, so that they're not prepared and they die. And then we have fewer mosquitoes the next spring and less West Nile virus. Okay, so now that I've introduced you to diapause and why I think it's important, we're going to start off on the first chunk of the research portion about how I think mosquitoes might be measuring day length and figuring out what time of year it is and initiating diapause, and specifically whether the circadian clock is involved in this process. So just to give you a bit of background, we have circadian clock genes, mosquitoes do too. These are the genes that regulate all of the daily behaviors in animals and plants and other animals. So in animals specifically, when they're feeding and when they're resting and when they're mating and all of that good stuff. And because they're so very important, they're found in photosynthetic cyanobacteria all the way through humans. And there's quite a lot of homology between the clock genes in animals, so that the same clock genes in fruit flies and mosquitoes are highly similar to the circadian clock genes that are in you. And much like everything else in molecular biology, the circadian clock was first and best studied in the lowly fruit fly, Drosophila melanogaster. So what I have for you next is an animation model of how the clock works in insects. So the circadian clocks, central primary circadian clock cells are located in the brains of insects. They're a specific subset of cells, and when inside the nuclei of these cells, two genes called period and timeless are just kind of hanging out until two other proteins, aptly named clock and cycle, act as positive transcriptional regulators. So they bind to E-box promoter regions on those genes, and they recruit RNA polymerase to the transcription initiation site, thereby increasing the levels of period and timeless messenger RNAs. The period and timeless mRNAs are then translated into their associated proteins, and then they form a heterodimer and move back into the nucleus, where the period component, the period protein, inhibits the action of clock and cycle, thereby suppressing their own transcription and completing this core negative feedback loop of the clock. This whole process takes about a day to complete, a little bit longer in some insects, a little bit shorter in others. And the way that the clock is kind of reset and informed and exactly matches the 24-hour cycle is thanks to the action of an additional protein known as cryptochrome 1, which binds to timeless in the presence of light and then degrades it. So this was the core clock model in fruit flies that was kind of laid out and discovered in the early 90s. And for a long time, we thought that that was how the circadian clocks looked in all other insects as well. But we now know that most other insects have an additional gene called cryptochrome 2. And this gene is similar to a gene within our own circadian clocks. And unlike cryptochrome 1, it's light insensitive. It doesn't bind to anything or degrade anything. Instead, it is also regulated by clock and cycle. And then its protein can also interact with period and timeless. And cryptochrome 2 itself can inhibit clock and cycle activity. And since its discovery in monarch butterflies in 2007, cryptochrome 2 has been found in pretty much every other insect outside of the higher diptera, like fruit flies and other higher flies. And so in a lot of ways, the mosquito circadian clock is more similar to our own circadian clocks than they are to another dipteran species, fruit flies, which I think is kind of interesting. Okay, so I've told you a lot about diapause. I've told you a lot about circadian clocks. Why do I think the circadian clock might be involved in diapause? Well, the reason is we don't know precisely how mosquitoes are measuring day length, but we do know that they have this very sophisticated group of genes that's providing them with valuable information on the time of day. And therefore, the same mechanism might also provide mosquitoes and other insects with information on how long the days are and whether they've experienced a long summer day and it's time to go into baby making mode, or whether they've experienced a short winter day and it's time to go into diapause survival mode. And this hypothesis was first made by the German botanist Irene Brüning. And there is a lot of evidence that the circadian clock in plants is involved and does regulate their seasonal responses. But it's been more of a mixed bag in insects. In the late 80s, David Saunders was working with mutants of Drosophila that completely lacked the period gene. And these flies were completely arrhythmic. They didn't know what time of day it was. But he found that they were still capable of entering diapause in response to short days. So this suggested that maybe the circadian clock is not involved in seasonal responses in insects. However, as many of you probably are well aware, model organisms are not always model organisms. And as I just told you, the circadian clock in Drosophila is pretty different from the clock in most other insects. And also, fruit flies have a very wimpy diapause response. So determining whether or not the circadian clock is involved in measuring day length is not a good, like Drosophila is particularly unsuited for that kind of question. So that's why I wanted to figure out what might be going on in Culex pipiens, the northern house mosquito. So as a PhD student in the Denlinger lab, what I decided to do was to characterize clock gene expression in diapausing and non-diapausing mosquitoes. This is because usually the levels of period and timeless RNA kind of oscillate or change throughout the day. And I thought that it might be possible that clock genes would show altered expression profiles under different kinds of day lengths that could explain whether or not the clock might be involved. This is because in 2007, researchers were working with European hamsters, and they saw that when they went into hibernation, their clock genes completely stopped cycling. The messenger RNA levels stayed pretty flat and low. So maybe the clock was also involved in regulating diapause in insects. And then the other way that I wanted to evaluate this was to knock down the expression of clock genes using a technique called Rna interference. So kind of just remove those genes from the system and break the clock and see whether or not mosquitoes could tell a long summer day from a short winter day and appropriately coordinate their seasonal responses. So that's what I did, and I'm going to share some of that data with you. So first, this is an expression profile for the gene cycle. And this was done by Carla Gentili in the southern house mosquito, Culex fasciatus. On the y-axis, we have relative messenger RNA expression. And then on the x-axis, we have the time of day given in Zeitgebers or time givers, where ZT0 indicates where the lights turned off, lights turned on, and these open bars show when the lights are on, and then the closed bars indicate when the lights are off. So we can see that cycle is showing peak expression shortly after the lights have turned on in the southern house mosquito. So I wanted to measure cycle expression in non-diaposing mosquitoes that had been exposed to 16 hours of light and 8 hours of darkness. And what I saw is indeed, yep, cycle is peaking right around the time of lights on. So that was good. Then 1 week into diapause, I measured in 1 week old diaposing mosquitoes that were exposed to 8 hours of light and 16 hours of darkness. I measured cycle expression and again saw a similar trend where peak cycle expression was occurring right around the time of lights on. 1 month into diapause, cycle was still cycling. 3 months into diapause, cycle was still cycling. And then by the time, and then after the mosquitoes had been in diapause for 3 months, I took them out of that incubator into another incubator that was set to long day conditions with 16 hours of light and 8 hours of darkness at higher temperatures. And again, we see cycle is still cycling. So unlike hamsters, diaposing mosquitoes have functional circadian clocks. Next, I wanted to measure the expression of a different gene, period, which is, as we know, a negative circadian regulator. It inhibits the action of clock and cycle activity. So here again, we have data from Carla Gentili and Culex fasciatus mosquitoes, and we can see that in this case, period is showing peak expression shortly after the lights have turned off. And that is what I saw in my 1 week old diaposing or non-diaposing mosquitoes. We're seeing peak period expression right around the time that the lights have turned off. But 1 week old diaposing mosquitoes are actually showing peak period expression much later in the night, around Zeitgeber 16 or 8 hours after the lights had turned off, which was highly curious and weird to me. 1 month into diapause, we're seeing that same trend with peak period expression occurring pretty late at night. But by 3 months into diapause, we're seeing the expected expression profile for period, with peak expression occurring right around the time of lights off. And then after the mosquitoes had terminated diapause and were exposed to long days again, the clock reset itself and period is showing peak expression at lights off at Zeitgeber 16 like it should. Okay, so that to me suggested that period is probably playing some role in measuring seasonal time, possibly, I don't know. So I decided I wanted to knock it down with RNA interference. And that's what I did. So I injected mosquitoes with either beta-galactosidase double-stranded RNA, which is a gene that they never make at all. So that was my control, or either double-stranded RNA that was specific to the period gene and should knock it down and prevent them from making the period protein. And I was able to successfully knock it down. And then I decided to look at 2 different phenotypes to see whether or not the mosquitoes were in diapause. And they were egg follicle length and fat content. So here's the egg follicle length data here on the y-axis, and we can see under long day conditions, knocking down period really didn't have much of an effect. Almost all of these mosquitoes had large egg follicles and were in a non-diaposing state. However, under short day conditions, my control mosquitoes were in diapause. Most of them, 84.6% of them, had small egg follicles indicating that they were in diapause. But when we knocked down period, we saw a significant increase in follicle length, suggesting that they had averted diapause, which was very exciting. Then when we look at fat content, we can see that under long day conditions, both the control mosquitoes and the mosquitoes that were treated with period double-stranded RNA had relatively low levels of fat. But under short day conditions, knocking down period prevented those mosquitoes from accumulating the fat that they needed to. So the controls had lots of fat, the period injected individuals had low levels of fat. So taken together, these results suggested that knocking down period prevented mosquitoes from entering into diapause in response to short days. So I tricked them. That was good. I also became interested in looking at another gene called pigment dispersing factor. So this gene is expressed in circadian clock cells within the brains of the blowfly Protoformia terranova. And work done in that species showed that the cells that make pigment dispersing factor are physically connected to regions of the brain that themselves are connected to the neural hemal organ that produces juvenile hormone. So this was a very nice way to link the circadian clock to the hormonal regulators of the diapause response. And so I wanted to determine what would happen if we knocked down pigment dispersing factor. Would that also prevent mosquitoes from telling what time of year it was? And fortunately, I was able to knock it down with double-stranded RNA. So here again are the control mosquitoes are shown in black, and the ones that were injected with PDF double-stranded RNA are shown in white. And we see a somewhat different trend, which is that under long day conditions, knocking down pigment dispersing factor caused mosquitoes to have significantly smaller egg follicles, such that 40% of the individuals that I measured were in a diapause state, which was very interesting. But knocking down pigment dispersing factor did not affect egg follicle length under short days. And then when we look at fat content, we see, again, knocking down PDF under short day conditions didn't have an effect. But when we knocked it down under long day conditions, those mosquitoes became very, very fat. So taken together, these results show that knocking down pigment dispersing factor causes mosquitoes that are reared under a long day, summer-like conditions to enter a diapause-like state, which is very exciting. So just to summarize, in a nutshell, the work that I did as a PhD student, I show that the clock continues to cycle throughout the diapause program. And early in diapause, period, which I showed you, and then also timeless and cryptochrome 2, all had this weird expression profile where they were peaking later on in the night, which initially perplexed me. And then I dug through the literature, as any good grad student should do. And what I saw is that I came across this paper by Mary Bowen, and she was measuring sugar feeding activity. And this is in non-diaposing mosquitoes. And you can see that for those non-diaposing mosquitoes, they're primarily feeding on sugar early on in the night, and then after that, they kind of settle down. But the diaposing mosquitoes are feeding on sugar all night long. So I'm now hypothesizing that the altered expression profile of period, timeless, and cryptochrome 2, which were peaking later in the night, might drive or get the mosquitoes to feed on sugar all throughout the night. And we also saw that knocking down any of the negative circadian regulators, which I showed you period, but the results were the same for timeless and cryptochrome 2, prevented mosquitoes that were reared under short day winter-like conditions from entering their overwintering diapause. And then kind of conversely, when I knocked down pigment dispersing factor, that tricked mosquitoes that were reared under long day summer-like conditions and caused them to enter a diapause-like state, which was very cool. So I now think that circadian clock genes are likely involved in measuring day length and regulating diapause, so I feel fairly confident replacing that question mark with a check mark. And currently, my lab is working on figuring out how exactly the circadian clock is connected to the hormonal regulators of diapause, including juvenile hormone and insulin signaling. So this is a fun thing that I get to do, is think about how this might all work. So over here, we have the kind of the core negative feedback loop, much simplified, right, with cycle regulating period expression and then period protein inhibiting cycle. Cycle actually also regulates another gene called pardoline protein 1, PDP1. And this gene is a transcription factor itself, and we think that it is regulating the expression of cycle. So kind of adding a secondary feedback loop into the clock. And based on kind of our results that when you knock down period, you get a long day response, that suggests us that cycle is itself involved in regulating long day reproductive responses. So we think that cycle might be involved in leading to an increase in juvenile hormone, which of course leads to egg follicle development and reproduction. And we also think that cycle might be regulating genes that are involved in insulin signaling, which helps keep the mosquitoes skinny. In converse, we think that partamine protein 1, in addition to regulating targets inside of the circadian clock, we think that it also might act outside of the clock to inhibit insulin signaling, and thereby indirectly, the inhibitor of my inhibitor is my activator, so indirectly activate FOXO and lead to diapause phenotypes. Okay, so we really wanted to figure out what's going on with PDP1. And I had an amazing undergraduate student, Vivian Chang, who basically repeated all of the work, or a lot of the work that I had done as a graduate student. So she dissected out over 600 mosquito brains and isolated RNA from them and measured the expression of PDP1 in diapausing and non-diapausing mosquitoes. So under non-diapausing conditions, PDP1 is cycling with peak expression occurring a little bit before the lights had turned off. However, under short day diapausing conditions, PDP1 is not cycling at all. It's pretty much flatlined, and the overall expression level is significantly higher. So that would suggest that PDP1 could be regulating diapause. Vivian also knocked down PDP1 with RNA interference, and what she saw is that it didn't really affect egg follicle length. So both the controls and PDP1 injected individuals under long days had big egg follicles, and under short days, they all had small egg follicles. However, knocking down PDP1 did affect fat content, such that once it was knocked down, it prevented females that were reared under short day conditions from accumulating all of the fat that they should have to be able to survive the winter. So I'm very happy that I recently got an Nsf Grant, which I'm so thrilled about. So it will allow me to answer some of these questions in more detail. And this is the model we proposed in the grant, that cycle is regulating targets within the circadian clock under both long and short days. But under long days, it's activating allodytropin, which is the pre-hormone that stimulates the production of juvenile hormone. We also think that cycle is upregulating pigment dispersing factor itself, which I showed you before, which leads to increased insulin signaling. And then we also think that cycle is involved in regulating a lot of other genes that are involved in making the mosquitoes reproductively active, but it's only able to do this under long day conditions when period isn't constantly inhibiting its action. Conversely, we think that PDP1 is acting to regulate diapause related genes under short day conditions. So under both long and short day conditions, we think PDP1 is again regulating clock targets like possibly cycle. But then under short day conditions, we think that it's regulating this gene called takeout, which is an inhibitor of juvenile hormone in Drosophila. And we think that PDP1 might also regulate a gene called SUSI, which is a known inhibitor of insulin signaling in Drosophila. But also we think that PDP1 might regulate a lot of these other genes that are important for generating diapause, but it's only able to do so under short day conditions. So we're going to test this by making cycle and PDP1 null mutants using CRISPR-Cas9, and then also using a technique called chromatin immunoprecipitation followed by deep sequencing, which basically allows you to take a transcription factor that's found in DNA and pull it out, and then get it to release the DNA, and then you sequence that DNA so you can identify all of the targets that it's regulating. And the reason that we're doing that is we're hopeful that there would be some specific targets that cycle is regulating to induce reproduction that we could then target with new control methods, which would be great. And then similarly, that PDP1 is regulating some diapause-specific targets that are unique to Culex pipiens that we could, again, develop novel controls for. Plus, I think it's really cool to figure out and identify the complete pathway from photoperiod perception to the generation of the complex phenotypes that are associated with diapause. And it would be the first time that anyone's done that in any animal. So pretty exciting. Okay, so thanks for bearing with me through all of that molecular physiology stuff. Now I'm going to get into some more practical, fun ecology things. So thinking about the ecological adaptations of studying diapause, and specifically thinking about we're trying to determine when Culex pipiens mosquitoes are initiating and terminating diapause in the field. And then also thinking about how anthropogenic influences like increased temperatures in urban heat islands, as well as artificial light at night, might be impacting seasonal responses in mosquitoes. So let's talk a little bit about diapause initiation. So as I told you before, West Nile virus is clearly a seasonal problem, and this graphic is taken from the Ohio Department of Health. And you can see that the number of West Nile virus cases is kind of peaking in late summer and early fall, but we do see West Nile virus onset well into October and even November, which is interesting because this is the period of active mosquito surveillance in the state of Ohio. It starts on May 15th and then ends on September 30th. Really, it kind of ramps down in August. And that's because mosquito control districts rely on amazing undergraduate students, and then they have to go back to college in August. And so there aren't so many people out there looking for mosquitoes. And we also don't know when mosquitoes are initiating diapause in the field. We think that it's probably sometime around October and November, but we're also still seeing cases of West Nile virus, so that's interesting. And we also have no idea when they're terminating diapause in the field, possibly in late February, March, maybe April. So that's what we're trying to figure out. This is data that I found for seasonal abundance of Culex mosquitoes in California, and you can see that, I hope you can see that the levels of mosquitoes are at their lowest level in the spring, like in April and May, right? Except for all the most southern extremes of California where, the summer gets really hot and dry. And I think this trend is going to be true in Columbus, Ohio, and in other parts, St. Louis, Missouri as well. And that it might be possible that if we have some really targeted and well-timed applications and control measures and interventions early in the spring, we might, hit our enemy at its weakest point and then thereby mitigate or prevent a lot of problems with mosquitoes and mosquito-borne diseases later on to the season. So this is where my amazing graduate student, Alden Dispersing, enters. He's been collecting mosquitoes from 5 different sites around Columbus, Ohio, that are also actively surveilled by Columbus Public Health and have a lot of mosquitoes. And he's been surveilling them and collecting mosquitoes throughout the fall, winter, and early spring. And he's been using 3 different methods. The first is a gravid trap where you put out some stinky water, really does smell awful, and then it's attached to a fan and a little trap up here. So the idea is that non-diapausing mosquitoes that are ready to lay eggs would come down here, try to lay their eggs, maybe succeed, and then get sucked back up into the trap. We're also using attractive sugar-baited traps. So these will be baited with nonanol and octanol, which are compounds in floral cues that Culex mosquitoes like. And that should collect both diapausing and non-diapausing mosquitoes, as well as highly sophisticated resting traps. They're not sophisticated at all. It's a cardboard box that's painted black, that the mosquitoes will go in to rest, and then you just kind of aspirate the mosquitoes out. So he's doing this throughout the fall, winter, and spring, and then taking the mosquitoes back to the lab. He's doing this as we speak right now, actually, and identifying them to species using morphological markers under a microscope, as well as PCR-based assays. And then he's going to determine whether or not they're in diapause by dissecting ovaries. So if they have really large egg follicles that look like they're about ready to be laid, this would be a gravid female, right? So she would be in a non-diapause state. This female would also be in a non-diapause state because she has large primary egg follicles, and also these are the trachea or tracheoles in her ovaries. And this, because they're nice and straight and broken off, that means she's parous, or she's laid a batch of eggs before. These are ovaries in a diapausing females. And you can see that the egg follicles are quite small, but also the tracheoles are not broken off. They're very curly, which indicates that this would be her first batch of eggs ever, and therefore she'd be more likely in diapause. Okay, so Alden clearly is working on that. He's like at step 1 of this process, just identifying the mosquitoes to species. But I do have his data on when and where the mosquitoes were around in Columbus, with some hiccups thanks to COVID. So he started sampling on the 27th of September, And we last collected mosquitoes in the fall on the 22nd of November. There were 6 mosquitoes total among all 5 of his traps. He collected his first mosquito on March 14th. And then 2 weeks later, he got up to 87 mosquitoes. And then a bit later on in April, he was up to 200 mosquitoes across, or over 200 mosquitoes across all of his traps. So the mosquitoes are clearly breaking out of diapause and are coming back long before we're actively looking for them, which is, interesting and something good to note. And hopefully we can figure out when they're actually going into diapause in the field and terminating it in the spring through more field sampling. So now I'll talk to you a little bit about some work that we're doing to figure out whether increased temperatures in urban heat islands might be interfering with seasonal responses in mosquitoes. And this work is being done by my graduate student, Lydia Fye, who's co-advised by Dr. Mary Gardner at Ohio State. Mary's an amazing lady. She does a lot of urban insect community ecology. And if you aren't familiar with her work, you should definitely check it out because she does super cool things. But many of you are aware that cities are warmer than surrounding areas, and that's because of the increased impervious surfaces in cities, streets, buildings, and the like, and also the pollution, which overall just kind of trap heat in a little bubble around cities. And previous work has shown that increased temperatures can delay diapause initiation. And as I told you before, increased temperatures are also the cues that mosquitoes use to know that spring is coming and to break out of diapause. So bearing this in mind, it's possible that the warmer temperatures in cities are prolonging urban residents' exposure to biting mosquitoes. They're around longer in the fall, and then they're coming out of diapause sooner in the spring, which would be not good. But we also know that higher temperatures cause insects to burn through their fat reserves much faster. And again, because insects are ectothermic organisms, this isn't surprising. This data is taken from cabbage white butterflies, and you can see that when the temperature is higher, basal metabolic rate is higher, which makes sense. So it could be that the increased temperatures in cities during the winter time would actually be bad for mosquitoes, and they would burn through their fat reserves, and there might be fewer mosquitoes in the following spring. So we will be doing we'll be doing some trapping and collecting to try to get at that question. But for now, Lydia has been doing a lab-based experiment to see whether or not increased temperatures really can interfere with mosquitoes' ability to go into diapause. So she's been rearing mosquito larvae under 11.5 hours of light and 12.5 hours of darkness, which corresponds to about mid-October in Columbus, Ohio, and then at 3 different temperatures, 20 degrees Celsius, 22 degrees Celsius, and 24 degrees Celsius, which we think correspond to, again, temperatures in mid-October in like more rural areas, suburban areas, and possibly in urban areas. Then once the mosquitoes emerge as adults, she's killing off some of them and measuring their fat, measuring the egg follicles and some others, and then keeping them alive and seeing whether or not they would bite. So far, all she's had a chance to do is measure their egg follicle size. And so here it is, egg follicle size on the y-axis, and then the different temperatures that the mosquitoes were reared at on the x-axis. And we can see that for the mosquitoes that were reared at the lowest temperature, most of them are in a diapause state with egg follicles less than 75 micrometers. But as the temperature gets higher, egg follicle size gets higher too. The really curious thing is that normally for mosquitoes that are in a non-diapause state, their egg follicles are larger than 90 micrometers. And we really don't see many mosquitoes ever kind of in this intermediate range, which is very curious. And I don't know whether or not these mosquitoes have decided to avert diapause or if they're in a quasi-diapause state. It's a bit murky right now. Hopefully, once she dissects some more mosquitoes and then we also measure their fat content and see if they're biting, that will give a clearer picture. Okay, and now for the final chunk, thinking about whether or not artificial light at night might impact seasonal responses in mosquitoes, which is something that I swear I came up independently of Katie. And the reason that we care about this is because over 50% or nearly 50% of the United States experience light polluted skies. So this is an image taken, that really illustrates how illustrates nicely how bright and how light polluted sight much of the United States is. And especially around different urban pockets, you can see really, really bright lights. And we also know there's been a lot of studies showing that artificial light at night affects the seasonal the ecology of insects that live in streams and fields. There have been a few studies that have shown that artificial light at night impacts the reproductive physiology of both mosquitoes and fruit flies. And then some other studies that have shown that artificial light at night disrupts behaviors in nocturnal insects such as moths and lightning bugs. But whether or not artificial light at night is impacting seasonal responses has not been very well explored, apart from Katie and now us. And the reason we think that this might be a problem is because we know that day length is the cue that mosquitoes are using to determine what time of year it is. So it's possible that the addition of light at night might trick mosquitoes into thinking it's a long day so that they become reproductively active, develop their egg follicles, bite us, and then lay eggs, which would not be good. So, and then, yeah, the cycle of life would continue. So what Lydia has done is she's reared mosquito larvae under, again, short day conditions, 11.5 hours of light or 12.5 hours of darkness at 20 degrees Celsius in incubators that either did not have artificial light at night, that would be our control, or in an incubator that had a very low level of artificial light at night, which is 4 lux. So that's about it's very dim. It's about being, I think, 10 feet away or so from a street lamp. Then once those mosquitoes emerged as adults, again, she was measuring their fat content, egg follicle size, and whether or not they were biting. So this is the data. We do see that mosquitoes that were exposed to artificial light at night had less fat than the control mosquitoes that were very nicely in a diapause state. The vast majority had enough fat that we would characterize them as being in diapause. But still, a lot of those mosquitoes that were exposed to artificial light at night had still quite a high level of fat, enough that we would characterize 70% of them as having enough fat to be in diapause, which is very curious. We also see that artificial light at night stimulates ovarian development. So under, this is egg follicle length, the control mosquitoes all had very small egg follicles, mostly under 75 micrometers in length, whereas most of the artificial mosquitoes that were exposed to artificial light at night had egg follicles that were above 90 micrometers, such that only like 17.5% of them had egg follicles that were below our threshold for considering them to be in diapause. And probably worse still, exposure to artificial light at night stimulated blood feeding. So the mosquitoes in the control cages that were offered a blood meal did not take it. None of them did. They didn't bite. They really were in diapause. But about 27% of the mosquitoes that were in cages that had been exposed to artificial light at night would take a blood meal. And then Lydia later offered those mosquitoes oviposition water, and they laid eggs and they hatched viable larvae. So they are using the blood to make babies. They're not using it for some other weird metabolic purpose. So to summarize these interesting results, artificial light at night causes mosquitoes to exhibit truly abnormal seasonal phenotypes. And just this picture really drives it home. So this is a non-diaposing mosquito. She's skinny and she has big egg follicles. This is a diaposing mosquito. She's pleasantly plump and she has small egg follicles. And then this is a mosquito that's been exposed to artificial light at night. And she is pretty fat, right? Almost as fat as a diaposer, maybe not quite, and also has large egg follicles, which suggests that this could kind of be the worst case scenario. She can produce babies and the offspring that would then possibly grow up to bite us as well, but she also might have enough fat that she would be able to survive the winter without feeding on anything else. So we're planning on doing follow-up studies to see whether or not these mosquitoes can actually make it through winter. And then, of course, the potential impacts of this work. We know that mosquito populations are higher in cities and particularly in low-income neighborhoods. So there's a big piece of social justice there. We also know that the number of cases of West Nile virus increased in the late summer and fall. So by postponing diapause initiation, either through exposure to artificial light at night or the urban heat island effect, could be causing mosquitoes to bite urban residents in the fall when they're more likely to be infected with West Nile virus. And we're still, artificial light at night increases the West Nile virus titers in house sparrows, which are an important reservoir species for West Nile virus. So really combined, artificial light at night might be creating a perfect storm for disease transmission in cities. Okay, So to kind of put all together these more ecologically focused studies, mosquitoes are still abundant in Columbus, Ohio through October and November and start appearing pretty early on in March in Columbus, Ohio. And we see that increased temperatures that cities likely experience may cause mosquitoes to avert diapause in the fall, and also that combined with artificial light at night also prevents mosquitoes from going into diapause in the fall and to continue biting us. So the long-term goal is to collaborate with Katie and then also to work towards building a predictive model to determine when and where people are most at risk of being exposed to infectious mosquitoes, and hence when and where we should best direct our control efforts. All right, so now the final slide, some take-home messages. First, I want to remind you that the circadian clock in insects and in mosquitoes is likely what is translating photoperiodic information, or how long the days are, into the hormonal signals that regulate seasonal responses in Culex pipiens mosquitoes. Second, that human-mediated changes in the environment, such as urban heat islands and artificial light at night, may be increasing disease risk in cities. And finally, that understanding how diapause is regulated, as well as how environmental variables impact seasonal responses, could allow us to more effectively control mosquitoes and prevent disease transmission. With that, I would like to thank my collaborators, students, funding sources, Katie for inviting me, all of you for listening, and if you have any questions, I'm happy to answer them.
Thanks, Megan. Thanks for the awesome talk. So let's see. Our first question is, do insects coming out of diapause have to repair themselves? So this person is thinking about how freeze-thaw cycles can damage molecules as well as larger physical structures.
That's an excellent question. Yeah, so I should say that mosquitoes are not freeze tolerant. I don't know of any freeze tolerant mosquito species. So if they freeze, they die. But that being said, they are worse for wear when they come out of diapause. So they have spent the whole winter kind of burning through their fat reserves. So they do need to find a nectar source to replenish their energy reserves. And they do that usually before they bite a person. And then one of the key features of diapause is that they're kind of preemptively upregulating stress responses. So things like heat shock proteins, for example. So if it gets really cold and proteins start to denature, the heat shock proteins can bind to those proteins and prevent them from forming big aggregates and blobs inside of cells, which would be deadly to the mosquitoes. So that helps. And then also, like, if there has been any kind of cellular damage, the mosquitoes would certainly need to repair that. Which again takes energy. And there's a reproductive cost of going into diapause. So usually mosquitoes that enter diapause or that have gone through diapause produce fewer offspring than those who haven't, which, you know, again, kind of speaks to the costs of overwintering.
Thanks. What is the diapause phenology in the native range of Culex pipiens? And how does that impact your thinking about findings in cooler climates?
That is a great question. So Culex mosquitoes are native to Europe, but they've been in the US and North America for hundreds of years. So in the native range in Europe and in Russia, it's pretty similar to the phenology that we see here. There's this lovely book that's all about Culex pipiens mosquitoes, but it's out of print. I borrowed it a few times from Dave, but I'm trying to get my own copy. I'm trying to remember from those cities. Like they've gone into caves where they've found Culex pipiens mosquitoes that are in diapause and kind of document when they've gone out of caves. So there's a lot of really lovely work there. But I think it's fairly similar to here. I will say that for most seasonal responses, they're based on latitude. So the further north you go, day length becomes more extreme, right? Or I should say as latitude increases, not alienate our southern hemisphere friends. So as latitude increases, day lengths become more extreme, and then winter has an earlier onset. And so then, not surprisingly, mosquitoes that live in northern environments initiate their seasonal responses, go into diapause sooner than southern species in the northern hemisphere. So I think, but I don't know that no one has actually measured the relationship between latitude and the timing of diapause in Culex pipiens itself, but that would be really cool to do both in North America as well as in Europe, and see if like the changes, that relationship between latitude and when they're entering diapause is consistent on the two continents. I think I only answered part of that question. Was the other part about climate change? Or maybe that question.
No, that was it, I think.
Okay.
So a related question is, so in New Orleans, the Culex do go into diapause, I believe, because they stop biting. But when a warm day comes, they will come out and start biting. So what do you know about this sort of must be intermittent diapause trait that southern Culex have?
Yeah, great question. So something I should have said and totally didn't, oops, they would be mad, is that diapause is a pre-programmed response. So they're entering into diapause before the going gets tough, before winter arrives. Whereas all insects are capable of becoming quiescent for a time, which is another state of dormancy, but that's a temporary state of dormancy that's usually in direct response to an environmental stressor, right? So like you find a honeybee, you put it in the fridge, it's gonna stop flying around because it's cold, and then you take it out of the fridge and then it warms up and it starts flying around. So the southern house mosquito, Culex cuncta fasciatus, does not have a diapause, but I'm sure it would be quiescent on days when it's too cold to fly. They would also probably, hunker down in houses, that's the name, the common name, in basements. And then once it warmed up, they would come back out and bite people.
Okay. So it is in fact a temperate zone critter and depends on caves for overwintering?
Yes, it is a temperate zone critter. It is, like I said, distributed in North America, in Europe, and then also in the temperate zones of Africa, in Australia, and Asia. It's basically all over the world. And then Culex pipiens is becoming more urban. So in more natural environments, it did overwinter in caves. And then once people came along and they became a bit more anthropophilic, now they're using our man-made structures like culverts and sheds and basements and houses and things like that.
Have you measured gene expression in wild mosquitoes early and late in the diapause season?
I have not, but that would be super fun, and we should definitely do it. The one thing I do worry, so the challenge of measuring gene expression is that RNA is really, really fragile. And then of course, so you would have to be careful how you collected those mosquitoes. You would probably want to make sure they were alive, and then you froze them on dry ice, isolated RNA from them. But we could totally do it, and we totally should.
So if you, so some of your gene knockdown work, do you, when you're able to knock down those genes and change the diapause trait, do they actually have higher winter mortality?
We haven't measured that. We injected the mosquitoes on the day that they emerged as adults, and then looked for their diapausing traits just a week later. But I would imagine that if the mosquitoes hadn't been able to accumulate the same level of fat reserves, that they wouldn't be able to survive for very long. So yeah, all of these great experiments that we should definitely do. I love this.
So how do you envision transferring this knowledge that you're gaining about seasonal responses in mosquitoes and and how those are regulated by genes into control strategies?
That's a great question. Mostly the gene regulation stuff I'm interested in just for pure nerdiness, like understanding the pathway. But what I really am, like early on in grad school, when I said like, oh, if you knock down the clock genes, you prevent the mosquitoes from going into diapause, or you can trick them into going into diapause. And people are like, oh, great, then that's what we should definitely do to control mosquitoes. But the problem is that clock genes are super well conserved among insects and even in us. And so if we were to target those genes or like mess with them or interfere with them, then we would likely interfere with clock genes in other beneficial organisms, which would not be good. So what I'm hoping is that as we discover more of the pathway, that we might find a very specific gene just for mosquitoes, just for Culex pipiens mosquitoes, that we could target and then either cause them to go into diapause in the summer or prevent them from going into diapause in the fall, possibly by using RNA interference. They've had some success with that in beetles and other agricultural pests, where you just put like loads of double-stranded RNA out into the environment, and because it's specific for that gene, it will knock down only that. If you design it well, and your double-stranded RNA is only specific to that species and doesn't affect beneficial species, then it would knock down the gene only in your target species and then prevent them from doing the right thing. So that's kind of the hope for it. But admittedly, it is a lot. It's much more basic research.
Sure.
Hopefully we'll have some usable applications in the future.
How do you spread a bunch of double-stranded RNA into a field?
Yeah, great question. So we did some experiments when I was a grad student where like you could put it in the water with the mosquito larvae and they would take it up. We don't know if it was like if they were drinking it or if it was like coming in through their siphon, if they were like diving down into the water, or exactly how it was getting in, but it was. In the field, I've seen people planning, like using this for like beetle pests in forest and thinking about like somehow like pumping it into a tree, the double-stranded RNA, so it's like inside of the phloem that wood-boring beetles are feeding. I think this is probably high. And then that would knock down their genes. In agricultural settings, they've also been looking at like using like a nebulizer to just kind of like spray it out, like with kind of high pressure, and then possibly getting it in through caterpillars through their spiracles as they're breeding. So who knows if that might work for mosquitoes or not.
Do you think that different light colors would affect mosquito reproduction differently?
That's a very good question. Yes. So Lydia used a warm white LED bulbs. There's been a lot of work on like, what are the photoreceptors that are involved in measuring day length, and it appears that light in the blue spectrum seems to work better for insects than others, and like more UV light. They also respond to tell seasonal time. I think that, and you would probably know more about this, Katie, but I think like the lights that are more in the blue spectrum would actually be worse and be more likely to interfere with seasonal responses in mosquitoes and the ecology and behavior and other physiological aspects in insects.
So along the lines of that, the last question, what types of light were used in your Allen experiments? Were they LED versus high sodium, for example?
Yeah, so it was warm white LED diodes that Lydia constructed. Just 8 of them in like a very kind of close arrangement. And that's something that we've thought about, should we change, and would like to try as well, like trying different, spectra of light and seeing how that affects this response. And then also kind of playing around with the exposure, because these mosquitoes were exposed to artificial light at night throughout their whole development, which is admittedly very contrived. Like in the field, if they're in a larval habitat under a street lamp, okay, maybe they might be experiencing artificial light at night through olive larval and pupal development, but once they emerge as adults, they would probably fly away and find some, darkened spaces. So, that's another thing that we want to try to play around with.
Yeah. All right. Well, I don't think there's any other questions. So thank you again, Megan, so much. It was a real pleasure having you. And we appreciate you taking the time to come in and share with us today. So thank you.
It was truly my pleasure. Thank you, Katie. And thank you all for listening and for asking such great questions.
Now we sit here awkwardly. Okay. All right. She did it.
I stopped sharing my screen at least. Oh, and she stopped the live streaming. Yay!
Yeah. Mm-hmm. There's a second of us laughing.